Hello, everyone, and welcome to another episode of Emerge. My name is Stoyan Stojcev. I'm part of the Resident Biosciences team. And thank you for taking the time to join us today for this episode. Before we got going, I introduce Sergio, the usual housekeeping announcements. We are going to record this session and we'll make it available online via YouTube channel. So look out for that in the next week or so. Please post your questions in the Q&A section, or if you want to have a live discussion, you can use the raise hand icon and now activate your microphone, and we can have a bit of a chat about the work that Sergio is going to be presenting. So I have the pleasure of introducing Sergio Mosquim Junior. He's in his final year of his PhD, based at Lund University. And his research focuses largely on biomarker discovery using mass spectrometry proteomics, using breast cancer as the model. And he's also working currently in a core facility at Lund University, where he's helping out the local community with analysis, with proteomics analysis. Previously, Sergio was based in Brazil. He completed his biotechnology engineering degree at the São Paulo State University. So, Sergio, I want to first thank you for taking the time to present your work. And yeah, you can take over.
Thank you so much, Stoyan. So first of all, thank you for the introduction, for the opportunity to present here, to talk a little bit about my work. I'll just switch to a nice pointer here. Let's see, with a laser pointer. Yeah. So the plan for today, to give an overview, I'll first give a quick background about myself. Then I'll move on to talking a little bit about breast cancer and the ScanBe initiative. Then we will jump into the analysis of these ScanBe samples, breast cancer samples, followed by a discussion of some of the results that we've obtained in a more or less proof of concept study. And finally, I'll give some concluding remarks and describe what our plans are moving forward. So as Stoyan pointed out, I'm originally from Brazil, where I completed my bachelor in biotechnology engineering at São Paulo State University. During my final year of the bachelor, I had the opportunity to go to Uppsala University as an Erasmus Mundus exchange student, where I'm mostly focused on drug discovery and development and proteomics. After that, I went back to Brazil and managed to now start my, or in 2018, start my PhD position as part of the CanFaster program at Lund University, where I'm currently in my final year and supervised by Fredrik Levander. So moving on to breast cancer. So breast cancer is currently the most diagnosed form of cancer worldwide, representing around 12% of all cases diagnosed in 2020. It is the #1 responsible for cancer-related deaths in women, and it ranks 5th in terms of overall mortality. There are different treatment options available, such as surgical intervention, chemotherapy, hormonal therapy, and radiotherapy, which some of them are given in combination and so on. And the therapy decisions are mostly guided by clinical pathological criteria. So some advances led to the definition of the intrinsic subtypes in the early 2000s by Charles Peroux. And these subtypes were originally based on microarray data. And later they got condensed into a signature of 50 different genes, which are now known as the PAN50, which stands for Prediction Analysis of Microarray Algorithm. And there are key markers in these signatures, such as estrogen receptor, progesterone receptor, the human epidermal growth factor receptor HER2, and the proliferation marker Ki67. These subtypes, which you can see here, named lumino A, lumino B, normal-like, HER2 enriched, and basal-like, they provide information about prognosis and treatment response. And these PAN50 signature has been established commercially via the Prosigna test, where here in Europe, you get classification into one of these subtypes and as well as a risk of recurrence score. But the intrinsic subtype was not FDA approved. So in the US, you only get the risk of recurrence. So even though there has been these advances in further patient stratification, there are still challenges associated with breast cancer, mostly related to overdiagnosis and overtreatment. And that means that patients end up being stratified and receiving more treatment than they actually need. And that has cost associated costs both for the healthcare system as well as the patient itself. And another issue that we currently face with breast cancer is late recurrence, where around 25% of patients that pass the 5-year survival mark will still end up dying of recurrent disease in the coming 15 years. And these numbers are even higher for estrogen receptor positive disease. So given this scenario, there is a need for new biomarkers in the context of precision medicine or personalized medicine. So as part of the advances that the molecular classification of cancer or breast cancer promoted, there has also been advances in terms of large clinical cohorts, and the SCAN-B is one of them. So SCAN-B stands for Sweden Cancerome Analysis Network Breast. The project was launched in the autumn of 2010, envisioned by Professor Åkerborg. And it has a primary objective to develop, validate, and implement clinically beneficial tumor analysis in the routine healthcare setting for patients with breast cancer in order to improve their overall care, quality of life, and subsequently outcome. It is fully integrated in the clinical routine, where the around 25 to 30 samples per week are acquired, which represents a coverage of around 85% of all cases in the south of Sweden. So as of 2022, the study has more than 7,700 patient data from the transcriptome. And there are an estimated number of 20,000 enrolled patients overall. So to give an overview of how this SCAN-B initiative works, so the patients end up being enrolled, and blood samples are collected in conjunction with the samples required for surgical intervention. During surgery, a biopsy is collected that then gets sent to the laboratory, and you can see that here in the blue part, where these samples get processed both in terms of tissue microarrays, as well as extraction of nucleic acids and preparation for subsequent sequencing. And this is all registered in the INCA registry with clinical information in the National Quality Assurance Register. So in addition to the RNA-seq or the transcriptomics data that is collected, and taken together with the fact that new biomarkers are needed, this presents itself as an opportunity to investigate the proteome. So better classification points towards the use of proteins as predictors or as potential biomarkers, given that they're more closely related to the phenotype and the possible impact of post-translational processing on protein activity. So in order to investigate the performance of different proteomics method and assess their potential use as a biomarker discovery tool, we've developed a protocol for parallel analysis of the transcriptome and proteome of these ScanB tissue samples. So the objectives we had were to first determine which was the best approach, and this is to do with data acquisition by mass spectrometry, either via data-dependent acquisition, DDA, or data-independent acquisition, DIA, and then analyze or perform some functional analysis, analyze the potential use of this proteome data for biomarker discovery. So besides providing an added layer of information to the already available transcriptome data, in a publication by Mertens and collaborators, they managed to show that as far as the PAN50 classification goes, there's a quite good overlap in these different subtypes when it comes to proteomics and the PAN50 signature, as you can see here on the top. But when unsupervised clustering was performed on both proteome and phosphoproteome data, as you can see here in the bottom, then you can see that the PAN50 subtypes end up being skewed, where you've managed to recapitulate a good part of the basal-like tumors, as well as the luminal ones. But for instance, the HER2 enriched, which are here by pink, they are kind of spread across the different clusters based on proteome and phosphoproteome data. So this pointed out or supports the use of proteome for or proteomics for biomarker discovery. So to give you an overview of the basic proteomics workflow, you can have a look up here. So this is the basic bottom-up proteomics workflow. And it's, I assume that most people in the audience are aware, but or work with proteomics, but in case there's someone who doesn't, bottom-up basically refers to the fact that you start with proteins, and these proteins undergo a digestion step with the different proteases, and they get cleaved into peptides. And these peptides are what are used for tandem liquid chromatography, mass spectrometry analysis. And it's different from, for instance, top-down approach where the focus is the analysis of intact proteins. So with this in mind, we've managed to integrate into a pipeline to work together this can be samples. So as I mentioned before, the tumor sample or the biopsies are collected and they get processed and further sent for RNA sequencing. So they get DNA and RNA extracted, and what we end up as starting material is a flow through, which mostly contains the proteins. So, and then we move on to a sample preparation, which, given the number of samples, we've managed to do it in a Kingfisher Flex system. So it allows us to process 96 samples simultaneously. And these, this is prior to LC-MS analysis. So in this project, we've used the Kingfisher Flex, and as far as LC-MS analysis, we were using an easy nano-LC 1200 model, together with Q-Executive HFX. And we've acquired both DDA and DIA data. So just to give an overview of the project itself. So as I mentioned previously, the samples get sent for RNA sequencing, and we get the flow through, which we use for sample preparation. And then we acquire the data both in DIA and DDA modes. And this was essentially to benchmark the two. After we've established a protocol in terms of data acquisition, then we move on to more functional analysis, where we compare the proteome data with already existing transcriptomics data. And then further on with the gene set enrichment analysis, a decision tree as a feature selection tool, and unsupervised clustering. So focusing now on the first part. As I mentioned, we first start with the benchmarking of different acquisition methods. So we were interested in knowing first which method would produce the highest number of proteins, or would yield the highest number of proteins, and how many of these proteins would be common across all analyzed samples. And for this initial study, we had a total of 116 samples. And secondly, how do these protein intensities correlate with their corresponding transcript? And I think it's quite interesting to point out here that given the nature of this CAN-B cohort, we actually have matched transcriptome and proteomic data because we're processing the same tissue piece. So focusing on the benchmarking, we first calculated the number of proteins found in the different acquisition modes, as well as how many are found across all samples. So we've performed a DDA acquisition with MBR, and that stands for match between runs, both with and without match between runs. Then we use DIA acquisition in a library-free mode, and in a library-based mode, where we use a library based on data-dependent acquisition samples. So as you can see here on this table, by using a library-free DIA mode, we've managed to acquire data from almost 10,000 proteins, and 3,200 of those were commonly found across all of these samples. So that points to this method perhaps being the best moving forward into further expansion of the cohort and functional analysis. Then, as when it comes to the correlation with the transcriptome data, we first look at the overall correlation, as you can see here, and this is the correlation using all pairs that were found between proteins and transcripts. And we also investigated the correlation after removal of low variance proteins and transcripts. And that essentially means that for each independent data set, we've filtered, we've calculated the variance for the different proteins and transcripts, and then we filtered based on the median. So the 50% with the lowest variance was removed. And this is due to the fact that you might have proteins and transcripts contributing to noise, and as far as biomarkers go, likely something that is stable across all samples are not very discriminant. So it's an interesting approach to have these removed. And as you can see here again, when we perform this using a library-free DIA approach, we have slightly better median correlation scores, both for Spearman and Pearson. But again, this is also taken together with the fact that we have quite more number of pairs detected. So taking everything together, as far as our benchmarking, we've decided then to select the library-free DIA approach moving forward with these samples. And this is what we use for different functional analysis, which I'll describe in a second. So going back to the questions that we had for the benchmarking, so the library-free DIA seemed to use the highest number of protein groups, with almost 10,000 proteins in total and over 3,200 found across all samples. And this method was also the one which provided the highest number of protein and transcript pairs and overall best correlation with transcriptome data. Now moving on to the second part, where we focus more on functional analysis. Since we had clinically annotated proteomics and transcriptomics data from the same samples, then we wanted to investigate by means of gene set enrichment analysis if these different omics could contribute to complementary information. So for this purpose, we identified proteins and transcripts which were differentially expressed or abundant in the different subtypes. And those are, as you remember, the luminose A and B's, normal-like, HER2 enriched, and basal-like. And then we applied the gene set enrichment analysis to identify pathways which could be enriched in each specific subset for each specific data type. In addition to that, we've trained a decision tree model as a quick feature selection tool. And we were interested in seeing if this model would be able to select proteomic features which were of biological relevance. So moving on to the gene set enrichment analysis. This is quite a busy picture, but as you can see, for the general part, the pathways showed a similar trend across the different subtypes for the different omics. But there were, for instance, when it comes to these luminal tumors, which are characterized by being estrogen receptor positive, when you look into the estrogen response early and late for these luminal tumors, then you have a significant positive enrichment that follows the same trend. And when it comes to basal-like and HER2 enriched tumors, which are estrogen receptor negative, then you have a negative enrichment of this pathway that follows the same trend as well. But opposite to that, we also had pathways such as the epithelial-mesenchymal transition and angiogenesis, where, for instance, here in the basal subtype, you have an enrichment in the opposite direction for the different omics, or you have an enrichment in only one of the omics. For instance, here in basal and HER2, where you have an enrichment for a negative enrichment for angiogenesis. So this demonstrates that these different omics can contribute to complementary information. So moving on to the decision tree model we have. So in order to construct these models, we compiled a list of proteins that were differentially abundant in different comparisons. So we compared receptor status, like estrogen receptor positive and estrogen receptor negative, grade 1 and 3, lymph node status, HER2 status, as well as some comparisons between these PAN50 subtypes. And the model was trained to predict the subtypes. So these clinical pathological features like grade and immunohistochemistry data was added as a way of not overfitting the model as well, since you're inputting proteins that are differentially expressed in the different subtypes and predicting the subtypes, but also to try and make it more biologically relevant and close to the clinics. So the proteins which were identified were anterior gradient 2 and 3. And these proteins, I think for the most part, they are expressed in or overexpressed in estrogen receptor positive breast cancer, often associated with a poor prognosis. We also have ERBB2 and 4, which are the receptor tyrosine kinase ERBB2 and 4, or HER2 and HER4. And at least for HER2 is a well-established biomarker in breast cancer, and the measurement is its measurement is necessary in all breast cancer cases. But also both of them are FDA-approved drug targets. The next protein we identified was the chitinase 3-like protein 1, and this protein is implied in promotion of inflammation by a Th2-like immune reaction and promoting macrophage activation, tumor growth, and is pro-angiogenesis. And this is a potential diagnostic biomarker as the sera levels are increased in patients compared to healthy individuals. And the last protein is RRM2, or ribonucleoside diphosphate reductase subunit M2. And this is a prognostic indicator of poor prognosis in several cancer types. And it's linked to angiogenesis promotion via VEGF. So it has a negative correlation with survival. So these were the proteomic features that the model managed to select. And this model, I mean, we had a total of around 80 samples. So the training was performed using 70% of the data, and that was roughly 60 samples. And we also wanted to check how the model would perform in unseen data. So we used the remaining 24 samples to test the model, and this resulted in this confusion matrix, where you have the reference label for the subtypes and what the model predicted. So, for instance, if you take into account the basal-like samples, we had a total of 5 in this test data, and the model predicted 4 as being basal and 1 as being normal. But overall, the model accuracy was reported at around 67%, which is not bad. I mean, first of all, as I said, the focus of the analysis was feature selection, and we were aware that we had quite a limited number of samples with only 84 available for this model, and there were quite imbalances in the different classes. But we were still quite surprised with the results. So to recapitulate the functional analysis, the transcriptome and proteome data contribute to complementary information, and we managed to see that yes, that our gene set enrichment analysis show that there are pathways which are enriched in different directions, and other pathways which are only enriched in one of the omics layer, even though for the most part they agree. And can a decision tree model select proteomics features of biological relevance? And we also managed to show that our decision tree model indeed managed to select features which reflect different biological aspects of breast tumors, be it an inflammatory response or already a marker associated with poor prognosis and so on. So to conclude, we've managed to establish a new workflow for the analysis of clinical cohorts, or these breast cancer or the SCAN-B cohort. Our machine learning model requires more sample for further validation and optimization. We showed that the proteome data can indeed be complementary to transcriptome data. And we've shown that these findings can potentially enable a better understanding of breast cancer biology and assist in the development of precision medicine with the development of new biomarker panels. So this was our proof of concept study. So now, after this, what is coming? So we've concluded this study. So now we're interested in expanding it further. So we've just recently agreed to analyze almost 200 samples more, and we wanted to shift the focus into a more clinical focus. So having stronger collaborations with clinical oncologists to be able to establish questions of clinical relevance. Because as in this first study, the samples were primarily selected to represent these different subtypes. And as I mentioned in the beginning, these subtypes are based on microarray data or further PCR data. And in the clinical practice, more often than not, tests are using proteins, and it's still standard that you will perform immunohistochemistry for all samples. So we were interested in kind of skewing away from the transcriptome and focusing more on what is there in clinical practice, and try and come up with questions based on that. And since now we're focusing more on proteomics, then we're interested in acquiring both full proteome and PTM data, or more specifically, phosphoproteome data. And we've managed to now establish a protocol which is based on published protocols from Jesper Olsen's group at the Novo Nordisk Foundation Center for Protein Research in Copenhagen, and discussions with collaborators, including Stoyan, for instance. So with that, I would like to acknowledge all the parts in this project. My main supervisor, Fredrik, my co-supervisors as well, Mats Solin, Valentina Sino, Sofia Valdemarsson, and Magnus Jakobsson. The Department of Immunotechnology, our collaborators, Johan Wallon Christesson and Lisa Rydén, and the ScanB steering committee, as well as the CanFaster program for this opportunity. So with that, I thank you very much.
Thank you, Sergio. It was really a great presentation. I enjoyed it. I enjoyed it a lot. So maybe I could start the discussion and with a couple of questions. One of the things that caught my eyes is right at the end, you talked about the new collaborations with oncologists. And what would you say would be the convincing argument for the use of proteomics, given the high correlation with transcriptomics data? What do you think is kind of the most powerful features that you could manage to extract just based on the proteomics data? Is there maybe some kind of subtyping that you're seeing additional to the subtypes that are known?
I think, I mean, the correlation that we presented here is based on these molecular subtypes as well. And it was shown, I can maybe go back to the slide. It was shown as well that in this publication by Martins, that as far as proteomics and transcriptomics go, the data would recapitulate quite well the different subtypes. But when you were if you were to do unsupervised clustering with proteomics and phosphoproteomics data alone, then that wouldn't be the case anymore. And I think in addition to that, it's still a fact that patients receive overdiagnosis and overtreatment, or they have late recurrence. So despite different technologies being available, there's still room for better stratification. And I think this is where, not so much for clinical implementation in the sense of mass spectrometry, but for the possibility of developing kits or medical devices that would further assist in patient stratification.
When you're doing unsupervised clustering, do you observe any novel subclusters or novel clusters besides what is kind of characterized with the PEM50? I think we didn't really focus on establishing clusters solely based on the proteomics data, or we were focusing more on seeing if patterns were similar. I think overall, if we use, I think, for instance, all features, either in the proteomics data set or in the transcriptomics data set, I think we have a good overlap, as it's shown in this publication. If we move more towards the proteins which were selected for the decision tree, where we have comparisons of different subtypes and clinical pathological features, then we see that things start to be a bit different, but we didn't really look into those further because we knew that the sample selection was a limitation from the beginning. Okay, so maybe a little bit more on technical side. one of the challenges is working with this multi-omics analysis is that you need to have kind of methods and extraction conditions that are compatible both with RNA-seq and downstream protein analysis. What advice do you have in this regard in terms of being able to do this kind of common extraction, but then be able to also then separate the samples and do the two different types of analysis, RNA-seq and proteins?
I think there has been quite overall quite a challenge that we've faced, because most of the methods for protein extraction and cleanup prior to digestion, I mean, they tolerate some concentrations of different detergents and so on, but we've seen that the conditions are a lot harsher than we expected, which results in having lower yield than we initially planned for this proteomic data. But then it depends really on what sort of questions you have in mind. For instance, since we're doing shotgun analysis and hopefully only analyzing samples once, that still allows us to start with a very low amount of input material and get enough out for mass spectrometry analysis. I think it becomes a bit more of a challenge when, like now that we're moving towards analysis of phosphoproteome, where we require much higher concentrations compared to just a global proteome analysis. And I think there we've now spent quite some time in trying to optimize protocols that deal with this sort of harsh buffers or higher concentration of detergents. But I think we also, from sample selection, since this time we were involved in all the steps. In this initial study, we've used a subset of samples to calculate protein concentration, and from there we've just performed a regression analysis and came up with a way of estimating protein concentration based on the RNA concentration based on absorbance measurements on a nanodrop. So seeing these samples and seeing that a few ended up being lost due to having low concentration, for the second cohort of samples, we were able to establish kind of a harsh cutoff in terms of RNA concentration from those samples that we wanted to select. So we had a minimum cutoff in the hopes that we would have higher protein concentration in this flow through as well.
To understand you, just to confirm you, the strategy here was to have the tissue and subdivide it and then extract it with the optimal buffers, rather than you try and find it compromised in the buffer to work with both RNA-seq and proteins.
Could you repeat the question again? Sorry.
So when you started working with the tissue, was it initially you subdivide it and then you do protein extraction, RNA extraction, or would you use a common buffer to do cellularization of maybe of the RNA and then you pull aside the proteins? Was that?
Yeah, I think I mean this is something that is out of our control since it's implemented in the clinics. So I think it's a commercial kit that is used for DNA and RNA extraction. So we don't have control of what buffers get used there. But I think if we were to perhaps establish proteome or the proteomics and that proteome analysis of these samples as another step, it could be that, say, a precipitation step is added before during these initial stages. So then we get cleaner samples to work with and subsequently have higher yields.
Yeah, and I suppose there's always this compromise between being able to get the optimal sample versus the throughput, and this is something we discussed offline a bit earlier in terms of the learning curve when it comes to processing largest sample cohorts. What would you say were the biggest lessons learned from your side in terms of being able to deal with 100 plus batches of samples? Is there some things that you have to implement to improve the efficiency, and also going forward, what would you be doing to do even with a larger cohort?
I think the main challenge is probably realizing that every step takes a lot longer. Like, you read a protocol and you think that I can do that in a day, but all of a sudden, when you have to open over 100 individual tubes and pipette different amounts from every tube, then you're spending at least half a day just doing that, and that is one step of the protocol. But I think now, since we have also like a larger number of samples, we've managed to, or we shifted focus into what could be replaced in the protocol and further optimized to, for instance, minimize pipetting steps or transferring steps. So that comes in the form of, for instance, trying to stay away from tubes and focusing on different plates and plastics that can be used in different instruments. And that already, I think, speeds up things quite a lot.
Yeah, no, absolutely. So another question I had is about keeping an eye on the quality of the data. And you mentioned that some of the runs you had to obviously discard post-analysis. What are the criteria for kind of doing system stability or process stability now, monitoring the sample prep quality, and what are the criteria for excluding samples at the end for you? Is it number of identifications, certain kind of signal that you expect to get on average, or what is it in your lab?
I mean, I think the first step is in the lab is to take notes of everything, because potentially anything can become a batch effect in the end.
Yeah.
So making note of that or being aware of that is quite important. And I think, I mean, thinking of the whole study design in the sense of, for instance, since we're using the Kingfisher Flex, that platform is compatible with 96 samples simultaneously. So if we have over 100 samples, we have to split into different runs. So making note of those known batches of samples that run together. And when it comes to the mass spec analysis, I mean, we're constantly adding quality control samples in the mix just to see that in terms of signal, the system is stable. And with the different batches, we add a pool of samples that we that then become reproducible or become our standpoint or a reference point when the different batches go through.
Yep, makes sense. So yeah, I don't see any more questions. So I want to thank everyone for taking the time to come in and join us today, and especially you, Sergio, for taking the time to present. It was really great, and yeah, see you everyone next time for the next Emerge episode, and look out for this recording. It's going to come up online in the next week or so. Cheers, everyone.
Thank you so much.
Bye-bye.